So today I'm going to be talking about some of my PhD research looking at the hemodynamic response in stroke patients. The hemodynamic response is regulated by changes in cerebral blood flow, cerebral blood volume, and the cerebral metabolic rate of oxygen consumption. And these changes can be impacted by both neural components, vascular components, and the signaling between the neural and vascular components called neurovascular coupling. It's important to study the hemodynamic response in stroke patients because stroke is a disease of cerebral blood vessels. And any research that uses fMRI to study changes in activity in stroke patients needs to take into account altered hemodynamic responses when analyzing this data. Previous studies have looked at the hemodynamic response function in stroke patients, but these are largely restricted to case studies. So this is an example of a patient with cerebrovascular disease who had a stenotic internal carotid artery. When researchers asked him to perform a movement task in the scanner, we see that the primary motor cortex on the side of the patent ICA showed a typical hemodynamic response. In contrast, primary motor cortex on the side of the stenotic ICA showed a delay in the time to peak and a decrease in the amplitude of the response. This is not just the case in primary motor cortex. Another study has shown that in a aphasic stroke patient who had a lesion near Broca's area, there was also a delay in the time to peak and a decrease in the amplitude compared to a healthy control performing a similar task, the same task. So because these studies have largely been conducted in single patients, we wanted to study this more systematically. And we did this by recruiting 23 stroke patients with visual field deficits and following them for 6 months after their stroke in a longitudinal study, with the first study visit being conducted within 1 week of their stroke. We also looked at 4 healthy control participants, and we used functional MRI to investigate the hemodynamic response by presenting a full field flickering checkerboard image in a sparse event-related design, where the image was on the screen for 2 TRs every 14 TRs, and this was repeated for 2 runs. This is what the hemodynamic response looked like in an early visual cortex voxel of a healthy control participant when we presented the full field flickering checkerboard with this sparse event-related design. We see that there's a typical peak in the hemodynamic response occurring about 3 to 4 TRs after stimulus onset. And we wanted to quantify the time to peak for each voxel in early visual cortex. In order to do that, we took every trial and concatenated them so that we could take the average of these trials to get a trial average bold signal for every visually responsive voxel in early visual cortex. And we then calculated the peak of that bold signal by looking at the time to peak, the time that voxel peaked in TRs. And so we mapped that in early visual cortex. This is a map looking at the intact hemisphere of a stroke patient, and we see that the time to peak bold response occurs between 3 and 4 TRs after stimulus onset, consistently in voxels in this region. In contrast, in the lesioned hemisphere, we see this population of voxels that has a time to peak bold response between 8 and 12 TRs after stimulus onset. And this localizes to the region of the diffusion abnormality that we see in the clinical scan that was conducted when they presented to the hospital. So we wanted to look at the average time to peak bold response in the lesioned hemisphere and compare it to the non-lesioned hemisphere across all of our stroke patients. To give you an idea of what that time to peak bold response should look like, here is the average time to peak response in healthy control participants in the left and right hemisphere, and you see that there is no difference between the 2 hemispheres. In contrast, in patients with primary visual cortex lesions, we see that the time to peak bold response is increased in the lesioned hemisphere, shown here in orange, compared to the non-lesioned hemisphere, shown here in green. And this difference between the hemispheres was present at a time point that was less than 1 week after their stroke, and continued to be different at the 1 month and 3 month time points, and was no longer different at the 6 month time point. This shows us that stroke patients with frank V1 lesions have a delayed bold response in the lesioned hemisphere 1 week to 3 months after stroke. We next wanted to look at the shape of the hemodynamic response, and to do this, we plotted every voxel's trial average bold response and binned it by that voxel's time to peak response. So here you can see the shape of the hemodynamic response for voxels that had a time to peak response between 3 and 5 TRs after stimulus onset. And this widely resembles what we would expect in a healthy control participant. In contrast, we see that in voxels that have a delay in the time to peak response, there's this pronounced initial dip occurring instead of a peak, and that the peak is occurring much later. And we wanted to quantify in all of our voxels to see if the pronounced initial dip that we're seeing here was consistent in voxels that had a delayed time to peak. And so to do that, we calculated the proportion of voxels with a given time to peak that also had a given time to trough. And if we plot that as a heat map, you can see here that the time to trough and the time to peak shows two different populations. The first is the population of voxels with a time to peak bold response within the normal range of 2 to 4 TRs after stimulus onset, and a time to trough that's occurring after the peak. In the second population, we see that these voxels have a delayed time to peak, but regardless of the delay, they consistently have a trough appearing between 2 and 4 TRs after stimulus onset. This pronounced initial dip is likely due to oxygen consumption exceeding oxygen delivery, and is key because it suggests that the neurons within these voxels are metabolically active and visually responsive, but they're not receiving the oxygen replenishment at the same speed as voxels with a normal hemodynamic response. So this pronounced initial dip is present when there is a delay in the time to peak bold response. We next followed up our analyses with some control analyses to confirm that the effect that we're seeing is specific to the infarcted vascular territory. So I already showed you the case where we have patients with primary visual cortex lesions, which is an infarction in the posterior cerebral artery territory, and looking at the hemodynamic response in early visual cortex. We next followed this up with these same patients, a subpopulation of these patients who completed a motor task every time that the visual stimulus was on the screen. And this allowed us to look at the hemodynamic response in primary motor cortex, which is supplied by the middle cerebral artery, whereas the infarcted territory was the posterior cerebral artery. We looked at the average time to peak in motor cortex for the lesioned and the intact hemisphere in these patients with primary visual cortex lesions. We found that there was no significant difference between the lesioned and the intact hemisphere. Since this is a subpopulation of all of the stroke patients that we studied, we also looked at this subpopulation in early visual cortex and confirmed the same findings as before, that there was a delay in the time to peak bold response in the lesioned hemisphere compared to the intact hemisphere. We then followed this up with another control analysis looking at stroke patients with visual field deficits who had deafferenting lesions, or lesions to the LGN or the optic radiations, which are supplied by the middle cerebral artery. And then we looked at the hemodynamic response in early visual cortex, which is again supplied by the posterior cerebral artery. And then in this analysis, we found that there was no difference between the lesioned and the intact hemisphere and the time to peak bold response in patients with deafferenting lesions. And these are looking at early visual cortex voxels. This is in contrast to patients with Frank V1 lesions, where we see that there was significant difference, and this is the same data that I showed you before. So taken together, these control analyses show that the hemodynamic response abnormality is specific to the infarcted vascular territory. Now, given the wide range of deviation of the hemodynamic response that we're seeing in our stroke patients compared to what we would expect in a healthy control participant, we followed up our empirical studies with some simulations to look at the impact of an extreme delay in the bold response on a general linear model analysis. We did this by simulating responses that varied in their time to peak between 1 and 12 TRs after stimulus onset. And this is compared to a canonical response shown here in black that has this time to peak response of 3 TRs after stimulus onset. We ran a general linear model analysis on simulated data from both a sequential presentation sequence and a random presentation sequence. Here I'm showing you polar angle stimuli that are used in retinotopic mapping, but it's important to note that our findings in these simulations apply to any type of general linear model analysis that's conducted on a population of individuals that has an abnormal hemodynamic response. So we first quantified the results of these simulations by looking at the false positive rate, which we defined as any voxel where the result was significant, but the predicted stimulus preference was different from the ground truth. And when we looked at the stimuli presented in a sequential order, we found that any shift in the time to peak response that was more than 2 TRs after the peak of a canonical HRF showed a very high amount of false positives, hovering around 100%. When we followed this up by looking at the systematic error, which we defined as the difference between the result and the ground truth, and looked at its relation, the relation between that difference to the time to peak, we found that there was a linear relationship between the difference between the observed and the ground truth, and the shift in the TTP from a canonical HRF for the sequential stimuli. This is highly problematic because it means that if researchers are using fMRI to study plasticity in patients with an abnormal hemodynamic response, it may be the case that what they think is plasticity is actually just changes in the hemodynamic response from one time point to another. We followed this up with a similar simulation conducted with random presentation of the stimuli. Here we see that the false positive rate is less than that in the sequential, but it's still quite high at around 50%. But importantly, with randomizing the stimulus, there is no systematic error or no relation between the observed and the expected, the difference between the observed and expected response and the shift in the time to peak response. These findings build on prior simulations conducted by Aguirre and Handwerker, but they conducted their simulations looking at very small deviations in the time to peak, whereas we look at much larger deviations. So in summary, I show that the shape and timing of the hemodynamic response is altered in stroke patients for up to 3 months after stroke. It's likely that this is due to a local vascular disturbance and not neural dysfunction, since the pronounced initial dip that we see is suggestive of an increase in the metabolic rate of oxygen consumption that's not being compensated for by an increase in cerebral blood flow and cerebral blood volume. These abnormalities that we saw were restricted to the infarcted vascular territory, and we showed that when you use a sequential stimulus presentation paradigm, you're prone to systematic error in cases where there is a delay in the time to peak response. All is not lost for fMRI analysis in stroke patients, however, and we recommend that going forward, researchers use random stimulus presentation paradigm, and instead of using a canonical HRF in their GLM analysis, they use an empirically derived HRF. We show that you can empirically derive the HRF using a sparse event-related design, but there are other ways to empirically determine the HRF presented by other researchers in the field. With that, I'd like to thank my advisor and co-advisor and everyone who helped with patient testing and recruitment and simulations, my funding sources, and my lab mates. Thanks.
So, good morning, everybody. I would like to thank the organizer of this symposium for inviting me to be part of it. I will be talking today about neurovascular coupling in cerebrovascular disease and dementia. And I would like to thank the grant and the founding agencies for allowing us to do the work. So I would like to maybe talk about neurovascular coupling, knowing that neurovascular coupling is really used more and more in brain imaging techniques, and is using hemodynamic signals to actually map changes in neuronal activity. And one of the main imaging technology is the fMRI bold signal. We know that the neurovascular coupling actually relies on the afferent input that are targeting an area, that are releasing different neurotransmitters, different neuromodulators, that these afferents are actually integrated by population of cells into the target area. And that the neurovascular coupling response then also is actually requiring the integrity of the neurogliovascular unit. So we ask the question, what is happening to this coupling in conditions where we have a cerebrovascular disease, so when the vessels are actually affected, or when there is an amyloid beta pathology on board, like is seen in Alzheimer's disease, or when there is a modulatory system that is degenerating, again, like in Alzheimer's disease. So in order to basically assess or respond to these questions, mainly in the two first questions, we use mouse model of cerebrovascular disease or the amyloid beta pathology. And we actually use mice with cranial windows, either small chronic cranial windows over the somatosensory cortex or full cranial windows over the whole cortical area. And we looked at changes into the level of oxygen bound to hemoglobin in the blood using optical intrinsic signal imaging. And for the blood flow response in all of those induced by whisker stimulation, we look at several blood flow response using laser spectral contrast imaging or sometimes laser Doppler flowmetry. So what we will be discussing today is really the response to the hemoglobin oxygen level and the blood flow. And in addition to actually talking about the oxy and total hemoglobin or the induced CBF response, the maximal response, we will also be looking at the time to peak. This is a variable, a parameter that is important because it tells us how fast the system is able to actually respond to the stimulus. So we'll be looking at the maximal response as well as the time to peak. So in the first model, which is a mouse model of cerebrovascular disease. It's a transgenic mice that overexpress transforming growth factor beta 1, that we call TGF mice. So these mice are very interesting because they have a pure vascular pathology. They have vascular fibrosis, they have string vessels or degenerating capillaries over time, but there is no amyloid pathology, there is no cognitive deficit, there is no cholinergic denervation, there is a chronic hypometabolism with time, and as well chronic hypoperfusion that develop with time, as well as impaired cerebrovascular reactivity because they do have a vascular pathology. So when we look at these mice, and we wanted to look at them over time, and this is the work of Dr. Millet Bourourou here, and as you can see, the wild-type mice compared to the TGF mice, even at 3 months of age, and this is a small cranial window, you can see that the TGF mice respond even less, even at that very early stage of the pathology. And as we can see for the total hemoglobin or the oxyhemoglobin response here, wild type compared to TGF mice is less at 3 months, and as we will see is also less at 6 and 9 months. But I wanted to show this figure because it shows that even in the wild type animal, there is an age-dependent decrease into the response in the neurovascular coupling response. Now, looking at these different parameters and looking at the slope or the time to peak, depending on the ages. So if you look at the 3 months, as we already said, there is a decrease in the maximum response in the TGF mice, no change in the time to peak at that age. The cerebral blood flow response induced by whisker stimulation, whether it's measured by laser spectral contrast imaging or laser Doppler flowmetry, is decreased already at that age, but there is no changes in the time to peak. When we move on to 6 months, we can see even a further decrease into the TGF mice, which is shown here for the maximum response. And we start to have a delay in the response. The time to peak is longer. And when we look at the CBF response, again, through laser speckle or laser Doppler, we do have a decrease, more severe at 6 months, and we start seeing a time to peak difference. This is even worse at 9 months of age, where barely we see no response, very, very weak response in the TGF mice, and there is a delay in the time to peak as well. And that's similar for the induced cerebral blood flow response, with a delay in the time to peak measured in laser speckle and a clear decrease in the maximal response. This is very interesting because despite these massive changes in the hemodynamic signal, when we look at the electrophysiology or the neuronal activity with an electrode into the barrel cortex, and at that point we use 1 Hz to make sure we had every stimulus would generate a local field potential. And we quantify the amplitude between the positive and the negative first peak. And as you can see, through age, there is absolutely no change in induced neuronal activity in the TGF mice. And when we decompose the LFP into the different spectral band, we have no change in the alpha, beta, gamma, and theta band, except at 3 months for a small change in the beta band, maybe due to the fact that the mice are anesthetized and movement is restricted. But clearly, all the hemodynamic changes occur without any change in neuronal activity. And this is also translated with absolutely no change into behavior measured with spatial memory here in the Barnes maze, wherever at 3 months, 6 months, and 9 months, the mice is able to find the location, indicating that there is absolutely no change into this behavior. And as you can see, with time, they are able to find a place where to hide from the noise and the light. Then we looked at as a second question, what's happening if we have the amyloid beta pathology on board, as seen in Alzheimer's disease? And we use a very classic model of Alzheimer's disease, the APP-G20 line, that overexpresses the mutated form of the amyloid precursor protein, the Swedish Indiana mutation. These mice are very interesting because they reproduce the soluble species pathology, the increase in soluble A-beta species. They have plaque. They also may have CAA over time, plaques, or if you wish, A-beta deposits around the blood vessel. They will develop with time cognitive deficit, and they will develop with time cholinergic denervation. And they also have hypometabolism, chronic hypoperfusion, and impaired cerebrovascular reactivity. So what's happening to the neurovascular coupling response in those mice? So I'm going to present the work here of Dr. Aline Zimmer, and Aline is using full cranial windows, and she's using awake animal. The work I presented with TGF mice was on anesthetized animal. Now I'm showing awake animal. And I'm going to show 3 months. She's now analyzing the 5-month-old animal. But what we can see is already at 3 months, the APP mice already have a decrease into the whisker evoked sensory response in the whisker cortex, in the barrel cortex, as shown here with the HBO response. And when we quantify that, it is clear here for HBO, HBR, and HBT that there is a decrease into the response in the APP mice. And even at that very early stage, there is also a difference in the time to peak. So the animal response is slower in the APP mice compared to the wild-type animal. Looking at cerebral blood flow response with laser speckle, at 3 months, we didn't see a difference in the maximal response, but there were a slight delay in the time to peak already. And what is interesting is that despite these changes in hemodynamic signal, very, very early on, at 3 months of age, there was no deficit in cognition in these animals, as measured again in the Barnes maze, and also measured with the novel object localization test, whereby the mice were investigating the change in location of the object, similarly to the wild-type animal. So this is interesting because we can detect absolutely very clear hemodynamic changes in neurovascular coupling, but these animals are actually cognitively intact as detected in those 2 different types of tests for spatial memory and more of executive memory. In our last question, it is also basically pursuing in the Alzheimer's disease field, whereby we know that there is massive cholinergic denervation in the cortex of Alzheimer's disease patients. As shown here, those are terminals for containing acetylcholine, very numerous in young control, much less in elderly control, but even much less in the Alzheimer's disease patient. So Dr. Clotilde Lecru asked the question, what is happening if we try to mimic this pathology in animal models? So what she did is that she injected in the ventricle the cholinetoxin saporin that will actually result in, after 10 to 14 days, will destroy cholinergic terminals into the cerebral cortex, as shown here compared to the saline group, where we have about 70% decrease, which is totally similar to what is seen in Alzheimer's disease patients. Then she will actually stimulate the whisker pathway in these animals with the decreased cholinergic tone in the cortex, mimicking basically Alzheimer's disease. And she will look at what's happening to the whisker evoked cerebral blood flow response and what's happening to the neuronal response to whisker stimulation, as well as to the identity of the population of cells in the target area in the barrel cortex that integrate those afferents. So what she found is that in the saporin group, low cholinergic tone in the cortex, there was a decrease in the cerebral blood flow response to whisker stimulation. And this was actually accompanied by a reduced size of the cells that are activated in the barrel cortex, as shown here by CFOS activity or CFOS immunoreactivity, decrease in the barrel size, as well as rostrocaudal decrease in the full extent of the barrel in the sensory cortex. And even though there is a decrease in the amount of cells activated, it is the same population of cells that are activated, namely pyramidal cells that contain or not COX-2, as well as interneurons that would contain vasoactive intestinal polypeptide. Moving then to the electrophysiology, measuring neuronal activity through a deep electrode through all the cortex in similar mice or similar rats, with a decreased response because they have decreased cholinergic tone in the cortex, what we can see is that the neural activity is decreased into the superficial and the granule cell layer, whereby the local field potential is decreased in both type of layers, and the barrel cortex will actually be located about here. And this is interesting because when we decompose the local field potential in the different spectral band, what we see is massive decrease in the high gamma, low gamma, beta, and alpha band, but we see an increase in the theta band. And this is interesting because in Alzheimer's disease patients, there is this mismatch between the low gamma and the high theta band activity, which is recapitulated quite well in this model. And knowing that the blood flow normally is actually correlated with the gamma band, it's interesting to see that we have a decrease in the blood flow response and a decrease in the gamma band activity. So on that, I would like to conclude by saying that when we're looking at brain imaging that are using hemodynamic signal, we have to be very careful because the neurogliovascular unit that is required for generating this signal, if the endpoint, like the blood vessel, is damaged, like in pathology of hypertension, diabetes, vascular dementia, or if there is an inflammation in the brain with chronic activation of the astrocyte that are required in that signaling, or again, if there is amyloid beta around the blood vessels or floating around into the parenchyma, together with cholinergic denervation in Alzheimer's disease, whereby a very potent modulator of cortical activity and cortical processing is decreased, we will see changes in the brain imaging that in Alzheimer's disease patients may reflect a combination of a neuronal and vascular and glial problem. But if we're looking at a pathology which only has vascular problem, we may still see a deficit in pathology that will due to the fact that the vessel is unable to respond, despite the fact that the neurons may be totally active normally, like we've shown in the TGF mice. So it may be very difficult to decipher basically the neuronal from the glial and the vascular component of that response. So we have to be careful in the interpretation. On that, I would like to thank you for your attention and thanking again the people who performed their work. Thank you very much.
Hello, hi. My name is Jana Kainerstorfer. I am happy to be presenting to you today about neurovascular coupling changes in the context of intracranial pressure and traumatic brain injury. I'm an assistant professor at Carnegie Mellon University in Pittsburgh, and I am leading the biophotonics lab there. So in order to get started, let's talk about intracranial pressure for a second. The idea is that the brain is inside the human head, inside the skull, and there are really three different compartments: the brain, CSF, so cerebrospinal fluid, and blood that dictate what the pressure is inside the head. If one of the volumes is changing size, pressure goes up. So two dominant examples would be hydrocephalus, where there's an increase in cerebrospinal fluid, or traumatic brain injury and swelling. Both of those diseases and many others are associated with worse neurological outcome. And so intracranial pressure is associated with worse cognitive outcome. And so there's really a need to monitor intracranial pressure and then optimize pressure such that neurons are protected. And so there is a very old idea around that is called cerebral autoregulation, which says that whenever perfusion pressure, which is really defined as the difference between blood pressure and the cranial pressure, whenever perfusion pressure is within a certain limit, autoregulation is intact. So what that means is that the vasculature is able to compensate for any kind of pressure changes by active constriction or dilation to maintain blood flow. Clinically, this has been translated to something that is called optimum cerebral perfusion pressure, which clinicians usually define as some sort of correlation between blood pressure and intracranial pressure. And the details are really not important here, but what has been shown is that if in the context of traumatic brain injury, if clinicians are optimizing perfusion pressure by either draining fluid or blood pressure medication, that this optimum perfusion pressure does lead to neurological outcome that is improved in those patients. And so what we're saying is, well, if autoregulation and neurological outcome are related, can we say this in terms of, is autoregulation due to perfusion changes linked to neurovascular coupling? And can we take neurovascular coupling as then an indicator for intact autoregulation? And so, as I said, autoregulation is usually defined or described by means of blood flow, and is there a particular range of pressure that maintains blood flow? The problem with this idea for any disease outside of traumatic brain injury is that intracranial pressure that is needed to really describe this autoregulatory mechanism, intracranial pressure can only be measured invasively right now by, for instance, a pressure probe inside the ventricle or the parenchyma. On the other hand, neurovascular coupling can be done non-invasively, can be measured non-invasively, for instance, by electroencephalography and near-infrared spectroscopy, which are the two methods I'm going to be focusing on today. So the way we look at neurovascular coupling then is this very general idea of neuronal changes lead to hemodynamic changes, and the hemodynamic response function, which is the convolution product between neuronal changes and the HRF, then predicts the hemodynamic signals we can measure. And so we can, for instance, measure neuronal changes with EEG, electroencephalography. The hemodynamic changes can be measured with fMRI, or in our case, near-infrared spectroscopy. And so to give you an idea of how that looks like in humans, it's placing sensors on the head, so completely non-invasive. And so the question we're asking is neurovascular coupling, and in particular the hemodynamic response function, a function of perfusion pressure and therefore intracranial pressure? So really showing this again, the idea is if autoregulation is intact, and in healthy volunteers, this cartoon here, the convolution with the hemodynamic response function is very well established. And the question is, what happens outside the range of autoregulation such that blood flow is no longer maintained? And so how does that change the hemodynamic response function? So the way we do this is in form of an animal model. In this case, we're using non-human primates. And we're measuring blood flow and hemoglobin concentration with optical methods, which I'll talk about in a second. So it's NIRS and diffuse correlation spectroscopy, and neuronal activation with EEG. We're changing intracranial pressure by placing a catheter in the ventricles that is attached to a saline reservoir. We have an invasive intracranial pressure probe in the parenchyma. And changes in intracranial pressure are achieved by using this saline reservoir that is attached to a cannula. By raising the saline reservoir, we can put more fluid in the brain and therefore change intracranial pressure. And to give you an idea of how that looks like, we step up intracranial pressure very controllably over the course of many hours, while measuring blood flow and the neuronal changes. So the system we're using to measure hemoglobin concentration in the brain is near-infrared spectroscopy. It's a frequency domain near system at 2 different wavelengths. And that really, we're only analyzing the relative changes in oxy and deoxyhemoglobin based on light absorption. So optical fibers are placed directly on the skull of the animals. The instrument we're using to measure blood flow in the brain is diffuse correlation spectroscopy, which again uses a bunch of lasers. In this case, we're using a home-built system. And by looking at the decorrelation of the laser, so in this case, the temporal intensity autocorrelation function, we can measure blood flow changes in the brain. In this case, we're using an 850 nanometer laser. And so combine those 2 instruments at 2 different locations in the head, are placed on the skull and measuring continuously. To give you an idea of kind of the changes that we induce, so as I said, the height of the saline reservoir dictates how much fluid is in the brain, and so very controllably we can oscillate in the cranial pressure, which then does not translate into blood pressure changes, but hemoglobin concentration changes as well as blood flow. We had two aims, which was, first one was trying to figure out, can we measure intracranial pressure non-invasively to overcome the invasiveness of the ICP sensors? And the other one is looking at neurovascular coupling changes. So in the first part, as I said, we're changing intracranial pressure continuously over the course of several hours. And what is known from a clinical perspective is that the cardiac pulsation, the cardiac waveform in ICP of the pressure changes its shape depending on which ICP baseline there is. And so what we have done is we have taken the cardiac pulsation and blood flow, so therefore non-invasive measurement, and have analyzed shape changes. So to give you an idea, the curve on the left-hand side is the non-invasive measured blood flow, cardiac pulsation versus intracranial pressure, color-coded with intracranial pressure. And as you can visually see, the shape is changing depending on the pressure level. And so we are describing this by extracting the features of the blood flow waveform, and we're training a form of machine learning algorithms or regression forests to then use those features to describe intracranial pressure non-invasively. So we have done that with 5 animals. We've trained the algorithm and then extracted intracranial pressure non-invasively. So what you're going to see is on the x-axis is invasive intracranial pressure, so that's based on the parenchymal probe, versus non-invasive estimates based on our blood flow measurement. So hopefully all those dots are laying on the unity line, and we get about a 90% correlation, and better shown in the Bland-Altman plot. We have about, we can extract intracranial pressure with about less than 4 millimeter mercury uncertainty. So as I said, we want to measure autoregulation, so therefore the invasive, non-invasive intracranial pressure sensor, but ultimately we're interested in neurovascular coupling, and it's really the title of my talk, where we measure with EEG and near-infrared spectroscopy the changes. So the animal experiments we are doing is, so the animals are anesthetized with fentanyl, and again, we're changing intracranial pressure based on the saline reservoir. We have the optical NIRS probe for measuring hemoglobin changes in the visual cortex combined with EEG, and we're showing visual stimuli, in this case of rotating checkerboards in a typical block design. So we have about 5 seconds of visual stimuli of the rotating checkerboards plus about 10-second rest in between. And we're looking at then the visual evoked responses as well as the hemodynamic response at each of the cerebral perfusion pressure values that are driven by intracranial pressure. So we're stepping up the perfusion pressure, and just to give you an idea, this is then actually measured blood flow changes, an average of 5 animals, where you can really see the plateau of Lawson's curve, so intact autoregulation versus impaired autoregulation. And then what we measure is color-coded here, the visual evoked responses, the VEPs with the EEG, color-coded with CPP, so that's perfusion pressure, versus with NIRS, the hemodynamic response. And so what you can visually maybe appreciate is that when we step up perfusion pressure, the shape of the VEPs as well as the shape of total hemoglobin is driven by perfusion pressure. And so what we then asked was, well, does the hemodynamic response function change as a function of perfusion pressure? So the way we do this is we take the magnitude of the EEG signal and the hemodynamic response function at each pressure level, and we're fitting a double gamma function. So that's the HRF, the hemodynamic response function, and then evaluate on the measured signals. And so if we do this at multiple different pressure levels, and we take at a healthy perfusion pressure level, we measure the hemodynamic response function, we calculate the hemodynamic response function and evaluate it at each of the pressure levels. What we have found is that essentially the hemodynamic response function from a healthy pressure level does not fit the hemodynamic signals we measure at higher or lower pressure than outside the autoregulatory range. And so the way we evaluate this is by a distance parameter that says how similar are the signals, the recorded versus estimated ones compared to the healthy level. And as a group average, essentially what we have found is that the hemodynamic response function is similar at healthy pressure levels and does change as a function of perfusion pressure. So we can visually show that by fitting the hemodynamic response function at each pressure level. And what we have found is within this plateau, within healthy pressure range, the hemodynamic response looks very similar. Moving outside, what we start seeing is shape changes in the hemodynamic response function. So again, visually, that's a healthy level versus unhealthy, not autoregulated values. And if we overlay this, what we have essentially seen is that when blood flow is maintained, the shape of the hemodynamic response function stays constant. Outside the autoregulatory range, the shape of the hemodynamic response function changes, therefore showing that there are indeed neurovascular coupling changes as a function of perfusion pressure. So in conclusion, we can measure intracranial pressure non-invasively, therefore perfusion pressure. And what we've seen is that neurovascular coupling is indeed changing with intracranial pressure, or rather to be precise, with cerebral perfusion pressure, and it does go hand in hand with autoregulation. With that, I would like to thank my lab, especially the students who worked on this, really valuable collaborators, and all of my funding sources. Thank you very much.
Okay. First, I would like to thank the organizer of the symposium, Colleen, Yunjie, and Jana, for this chance to speak here. And my name is Yunjie Tong, and I'm from Biomedical Engineering Department at Purdue University. And my topic of the talk is assessing various hemodynamic response in hypercapnia using functional MRI. So different from other speakers in the symposium, my focus is on the hemodynamic response to vasoactive stimuli, right? Which is really to the physiological stimuli instead of to the neuronal activation. So the mostly widely used vasoactive stimuli is CO2 challenge. So in which people breathing elevated CO2 to cause hypercapnia. Then because we know CO2 is potent vasodilator, so that would dramatically increase the blood flow. So that's why CO2 challenge is always considered as a stress test to the brain, and because it can expose some hidden perfusion deficit in the brain, especially some brain disease such as TBI. And however, the CO2 challenge is a very complicated process, right? So first, the CO2 has to be delivered to the brain by blood, right? So the blood will carry the CO2 to different brain voxels at different delays. And secondly, when the CO2 arrives at different voxels, and the voxel will have its own hemodynamic response function to the CO2, right? Because that also depends on the position of the voxel, what kind of blood vessel surrounding the voxel, what kind of tissue surrounding the voxel. So that will decide the hemodynamic response for the voxel, which is quite different. I'll show that later. And the last thing is the magnitude of the regional response, right? So this actually is the mostly used parameter in the CO2 challenge. It's called the cerebral vascular reactivity. It is defined as voxel-wise percentage Bose signal change per millimeter mercury change in the arterial partial pressure of CO2. So I try to move this. Okay. So here I'm giving you a typical CO2 challenge protocol. That in this protocol, subject was breathing 2 blocks of elevated CO2, which is about 10 millimeter mercury higher than the subject baseline. And at the same time, we measured end-tidal CO2. Right? You can see on the right, that's the end-tidal CO2 measurement. And in this particular case, and generally speaking, the end-tidal CO2 is a very good surrogate signal for the arterial partial pressure of the CO2. Okay, so this is the one subject's example data. You can see I randomly pick up three Bose signals from the subject, and I'm showing the waveform of these 3 Bose signals. They look quite different, right? So the reason why they look very different is because they have very different hemodynamic response function, HRFs, as I mentioned before. So for the rest of the talk, what I would like to do is to show how the difference in HRFs can impact the calculation of the CVR, can impact calculation of the delay maps. Okay, first let's talk about the delay maps, right? So the delay maps, as I said before, is really the way to assess the timing of the CO2 to arrive at different voxels. So the simple way to do that is to do a cross correlation between end-tidal CO2 measurement with every Bose signals in the brain, right? So then you can calculate the delay using the end-tidal CO2 as a reference point. Then you can calculate the delay for each voxel. And here is the typical delay map we get from the CO2 challenge. You use this method. You can clearly see that the pattern of the delay map is very clear. That makes a lot of sense given the blood vasculature. And also, it's very symmetrical. It's beautiful. And the time of the delay range from -10 seconds to +10 seconds, which is a little bit long. When you look at the histogram of this delay map, you can see it's Gaussian distributed. However, the range of the delay value is go from -15 seconds to 15 seconds. This is not right? Because we know the transit time of the blood through the brain is about 5 seconds in resting state, right? So, and the CO2 is vasodilator. So as a result of the vasodilation, you have increased blood flow. So that will lead to shortened transit time instead of longer transit time. Especially in this case, it's 30 seconds versus 5 seconds is too long. So something's wrong. So the possible reason for the error is spurious correlation. And the 2 main contributors for this severe spurious correlation error is coming from, one is we use very, very low frequency oscillation. In this case, it's about 0.004 hertz. That's the CO2 modulation frequency. So it's too low. The second and most important thing is we underestimated the waveform of the Bose signal and its effect on the delay calculations. So I will illustrate my point using 3 simulated Bose signals, right? So in this particular case, I simulated 3 Bose signals using different HR function. So in this case, you can see 3 Bose signals showing here as the blue, red, and yellow. And you can see they share the same breaking point. That means, in the black circle, that means, the CO2 arrive at these 3 voxels exact the same time. The only difference between these 3 voxels, is their HRFs. In other way, is their, waveforms of Bose signal. However, in terms of the breaking point, is exact the same, right? So when I cross-correlate these 3 Bose signal with end-tidal CO2 measurement, I should get a similar value if I'm interested in the delay time. So the result is quite surprising. You can see that the delay time for these 3 simulated curves is going from -2 seconds to all the way to -21 or 22 seconds. So this dramatic difference in the delay time is not causing by the same breaking point. However, is really causing by the very different waveform of these three voxels. So how can we solve this problem? So the way to solve this problem is, instead of using very, very low frequency oscillation causing by the CO2 modulation, we use another frequency band signal. So we use what we call the low frequency oscillation signal. So in this case, low frequency oscillation is defined as 0.01 to 0.1 hertz. We can extract low frequency oscillation from the Bose signal by using the method I'm showing on the left. So you can first identify the CO2 modulated signal as a middle panel, then you subtract that from the original Bose signal. The rest of the signal will show as the low frequency oscillation signal. Then what you need to do is you just need to correlate the low frequency oscillation signal from each voxel with the global mean signal of the low frequency oscillation. Then you can get a very clear delay map for the CO2 challenge. And in this case, you can see the pattern of the delay map is similar to the delay map we saw before. However, the transit time or the duration is much smaller. In this case, it's only from -3 seconds to 3 seconds. It's way smaller than the previous one. To further validate my point, here I'm showing you the delay map generated from resting state compared to the delay map generated from the CO2 challenge. Both of them using the low frequency oscillation signal to calculate the cross correlation. You can see that in terms of the pattern, they look very similar to each other. Given the blood flow path is exactly the same, so they should be similar, right? So, here I'm showing the scatter plot between the delay map from resting state and the delay map from the CO2 scan. And if you see that, if these two delay maps are exact the same, then the dots should be scattered around the line of equality, right? Which is showing as the black dot line. However, the real fitting of the line, the real fitting of the data is showing the red line, so which has a bigger angle than the 45 degrees. So this actually indicates the blood flow in the CO2 scan is faster than the resting state, which is exactly what we expected because the CO2 effect on the blood flow. Okay, so here what I would like to say, talk about the impact of the HRF to the CVR measurement, right? So for each voxel, actually CVR can be measured from the scatter plot between percentage Bose signal and the untitled CO2 measurement. Now you can clearly see on the lower part of the figure, I'm showing you the black dots and the black line are black lines are the fitting for the black dots. Then the slope of the black line is our CVR values. However, in this case, that we use the same untitled CO2 to fit for the old Bose signal, which is not good because we all know the Bose signal change voxel by voxel, right? So the better way to do that is really to convolve untitled CO2 measurement with voxel-specific HRF. And you can see the result of this convolution I'm showing as the red curve in, together with the original Bose signal. You can see the red curve fits much better with the Bose signal to each individual voxel. So the new CVR can be derived, right? The updated CVR can be derived from the percentage Bose signal change and the scatter plot from percentage Bose signal change and the convolved untitled CO2 measurement, which is showing the purple dots and the purple line. And you can see for these two particular voxels, the original CVR value is showing in the black. They're about the same, right? So we cannot separate the CVR between these two voxels. However, when we use updated method involving the HRF for each individual voxel, that suddenly the CVR value become very different from these two voxels. So in other way, the method shows increased sensitivity. Lastly, I'm going to address the elephant in the room, which is the HRF itself is a very important parameter to assess the perfusion in the brain, right? So in this case, as talked by many other speakers in the symposium, so it's better for us to really have HRF map for the brain. So there's many ways to map the HRF in the brain. The way we did it is a very simple way. We generated a database for all the HF models for the CO2 challenge. Then we find the proper HF from this database for each individual voxel. Then the index for the HF can be used to generate the HF map. Right. So from this particular map we generated, you can see it looks very different from the delay map and the CVR maps from previous calculation. Then that means HRF map showing is very different, assess very different perfusion parameters. Not like the delay, not like the CVR. So to conclude that, so CVR, I'm sorry, not CVR, just HRF to the vessel-active stimuli are different from those to the neuronal activation. Secondly, the HRFs are important in the CO2 challenge for accurate delay assessment for the CVR calculation. Right? The last one is HRF to the CO2 challenge. It could be important primarily by itself, right? You can assess very different perfusion parameters other than the delay and the CVR. So I would like to thank my students to do the work, and I also like to thank my collaborators. And also these are the references I draw a lot of inspiration from to do this project. And thank you. And welcome for questions.